All right, ladies and gentlemen, welcome back. So we're going to continue talking about the structure of your cell membrane. We've talked about your phospholipids, we've talked about the cholesterol, we've also talked about the fact that proteins exist. Now let's talk a little bit more about what those proteins are doing. So your membrane really is a collage of a bunch of different proteins. Some of those proteins are connected with carbohydrates, so these glycoproteins. A lot of these are more tags for your cells to recognize what type of cell I'm dealing with. Um, is it my cell? Is it something else that shouldn't be there? Right? You've got glycolipids, which are a similar idea, so carbohydrates attached to those lipid heads. You've got all sorts of other fibers that are part of your ECM, your extracellular matrix, and then you've got a bunch of proteins that are directly embedded. You've got your integral proteins, which will go straight through, and then peripheral proteins as well. And most of those proteins are what determines the overall function of the cell membrane beyond just being some selective barrier. So when we look at transmembrane or integral proteins, these are the ones that penetrate all the way through the membrane. So they've got pieces that stick off on either side of the cell membrane. This does mean that they need to have both hydrophilic and hydrophobic regions. So the regions that are sticking outside of the cell and internal to the cell have to be made of amino acids that are hydrophilic, while we get the amino acids that are connecting here up against those fatty acid tails, those need to be hydrophobic. And then when we look at the internal structure of that protein, a lot of times these will be channels or pump systems. And so that internal structure is often hydrophilic as well. Now, keep in mind that there's a start and end to these proteins. And so this is just kind of a review of your basic protein structure. We've got that N terminus, right? This is where that amino group is at the end. And then your C terminus is your carboxyl end. So remember that there's directionality to the way that those proteins are formed and the way that they fold. So as a result, that's going to be pretty important in the way that they integrate then within that membrane, because if they misfold because of some sort of mutation, for example, they may not be able to embed in that membrane. And so whatever their job is may not be happening within that cell membrane because of the misfolding. So just something to kind of connect back to the biochemistry. Your peripheral proteins, these are ones that tend to be much more loosely bound to the surface of the membrane, more often than not to the internal surface. A lot of these might be connected to things like cytoskeletal fibers to help with cell movement and things like that. So you've got some major functions of membrane proteins. You should be familiar with them. I'm going to kind of give you some real basic idea here, but you should be able to define what the functions are and provide a specific example. So as you're working through the text, make sure you're spending some time here. We'll talk about some of these here specifically this unit. Some of the others will come back in later units, some of which we have already talked about. So you've got your transport proteins. Those will be the ones that we spend most of the time talking about here. Some of them are functionally capable of enzymatic activity, so aka enzymes. So remember what that means. Some of them will be involved in signal transduction, so being able to receive molecular signals from other cells and adjust what that means for that cell to be doing. We'll come back to more of that at the end of the school year when we get to cell communication. Some of them are part of intracellular joining, so being sort of those gap junctions, tight junctions, being able to seal one cell to the next in a tissue. Others will play roles in attaching cytoskeletal structures as well as fibers out in your extracellular matrix. And then that cell-to-cell recognition is also here as well. So again, here's just some more diagrams. Make sure you spend some time here, read through what these mean, know some examples, and again, we'll come across some more of them either here in this unit or as we continue throughout the rest of the school year. We have already talked about a few of them too, so make sure you go back and make those connections to units that we've already covered. A little bit about that cell-to-cell recognition here, and we'll talk more about it as we continue moving forward this year. Remember that those cells have to be able to recognize each other. You have to be able to identify one cell in your body from another type, also be able to recognize that it is in fact your cells. And a lot of that recognition comes from the carbohydrates that are attached to that plasma membrane. Either they're bound to those lipids, and so we call them glycolipids, or they're bound to those proteins, so we call them glycoproteins. So the carbohydrates that are on the external side of your membrane, meaning facing towards the environment, vary among different species. So that's how your body is able to recognize whether that cell comes from human or not. It also varies from one individual to the next. So being able to recognize your cells from someone else's, which does complicate things like organ transplants, as well as your body even being able to recognize different cell types within you. So being able to recognize a cardiac muscle cell versus a skeletal muscle cell versus an epithelial cell, so on and so forth. Another thing to just kind of keep in mind while we're talking about things sticking off of your membranes is that there is distinct sidedness to them. They have inside and outside faces, and this exists not only in your plasma membrane, but it's also true within the membranes that make up things like your endomembrane system, the ER, the Golgi, so on and so forth. So when we're moving things throughout that membrane system, the proteins that are synthesized in that endomembrane system need to be produced and put into those membranes in the right orientation, especially if they're going to end up being part of those membranes that end up as part of our cell membrane. I need to make sure that receptors and glycoproteins and all sorts of things are properly labeled and properly oriented. And so when we're going through those vesicles, I need to make sure that the orientation is appropriate. And a lot of that happens in your Golgi so that when they get here, they're on the right side of the membrane.